which I should start doing. I'm going to start doing that. And instead of giving them the names of like the dates, like September or whatever today's lecture, I'll just start giving them titles. All right, so I'm recording. All right, so that is like the cell membrane. That was a chapter about the cell membrane, that last one. This one is about the cell cycle. And really all it is, the cell cycle is taking one cell and dividing it into two cells. This one's going to be shorter. So we take a cell and divide it into two cells.
This is not on the exam coming up?
It is.
It is, okay.
This one's on the exam coming up, not as long as the other one. So how do we take one cell and divide it into two cells? So we have different, if we're talking about like cells dividing, we have different types of cells in our body. We have sperm cells and sperm cells and egg cells. They go through one type of process that we're not going to talk about. And then we have every other type of cell in our body, cells that grow. And so cells that grow, we're talking about what we call somatic cells. And I'm not going to test you on this word, but just so you know it. If you see the word somatic, it means like growth. So we're talking about somatic cells, all of our other cells. Red blood cells, those don't divide. Those are kind of, we make them, they live for like 4 months and then they die. So red blood cells are kind of unique. And sperm and eggs, they're unique as well. But everything else in our body splits from one cell into two, and then those two cells into four, right? And so there's a process, there's different stages that these cells go through. 3 stages. And the first stage is called interphase. And in interphase, what's happening is that you're getting the cell ready to split in half. So we need more organelles. And just to remind you, you should know that list of organelles. So, like ribosomes, Golgi apparatus, there's lots of websites that will like just quiz you on it, right? So just the organelles, there's like 8 or 9 of them, lysosomes, ribosomes, mitochondria, Golgi apparatus or Golgi bodies, rough ER, smooth ER. Like, what do they do? So if I say rough ER, you're thinking proteins. Smooth ER, lipids. Golgi, they package proteins. Mitochondria, they're like the ATP. Centrosomes or centrioles, they're involved in mitosis. So I've got them on those notes. It's like a Word document. They're all listed. But you really just need to know like just a couple of words about each one of those. But all these organelles that are in the cell, they're all starting to double. There's actually 3 phases to interphase. Not that it's important, but in G1 and G2, organelles double, or however you want to say it. There's like other ways to say that. I mean, I'm sure they're not exactly doubling, but they start replicating. You want twice as much mitochondria. You want twice as much lysosomes, because this cell's going to split. You can't split and have half as much ribosomes, because then when you split again, now you're going to be down to a fourth, right? And eventually, that's it. So you got to make some more. Got to make twice as many ribosomes, and then you're going to split in half. Then it'll be an equal amount. In the S phase, S stands for DNA synthesis. Remember, synthesis means to make. So in the S phase, we're making DNA. That's interphase, that's it. That's what happens in interphase. So by the end of interphase, you end up with a cell that's a little bit bigger and super crowded. It's twice as much stuff in that cell. And you look at the nucleus, that's where all your DNA is. The nucleus is like really big and packed as well. There's like twice as much DNA in there too. So it's like a cell that's just got twice as much, it's twice as crowded now. That's the first phase of the cell cycle. And actually, this is the phases of interphase. The second part of the cell cycle is mitosis, which you learned about in high school, but you probably forgot because you took the test. It's like one of the only things I remember from high school. Mitosis, mitose split. And I didn't know what it meant. If you say, what does that mean, mitose split? I don't know. It didn't get me through the test either. So mitosis is a story of the nucleus and how the one nucleus is going to divide into two. And there's different phases to mitosis. And what I'm going to do is I'm going to write a couple of things for each phase. So the phases are, I'm going to write 4 phases. Like some books will have 5, some books will have 4. I'm just going to go back to 4. It doesn't really matter. They're both correct, but I got to think of what it is. Prophase, Metaphase, Anaphase. Do you guys remember the 4th one from school?
Anaphase?
Got that one. What's after that? Why would you really practically need to know this? Only if you have to take pharmacology. So if you're going into nursing, you're going to take pharmacology. When you take pharmacology, you're going to have to know this. So I'm really kind of keeping it rather simple. And by the way, I guess I'll go through this and then I'll tell you the third one. And the third one's called cytokinesis, which just means the cell breaks into 2. Let's talk about all this. So here's a cell. And the notes have pictures, right? And there's really lots of videos. One website is called cellsalive.com. They run through all of this. You could learn this pretty quick. Here's a cell. Here's the nucleus. Inside the nucleus is like all the DNA. And then there's lots of organelles in here. For this particular chapter, there's only one organelle that we're concerned with, and they are called centrosomes, which I already started spelling wrong. I only had 1 letter in my centrosomes. And they're made from these things called centrioles. I'm telling you that because I use both words. So for this lecture and for your exam, centrioles, centrosomes, same thing, interchangeable words. Even though they're not technically interchangeable, but whatever. So you have centrioles, and remember this cell just doubled. So there's not one centriole, there's like two. So I'm writing them like, I'm making like little stars to represent the centrioles. So here they are. They're right next to each other. There's two of them. What they're going to do is they're going to start moving apart. So by the time I get to metaphase, they're going to be like here on opposite ends of the cell. Right now they're together. So here they're just starting to move apart. So I'll put, I'm going to put two things. One, chromatin condense. And then centrioles move apart. What does it mean, chromatin condense? I'm going to show you two nucleuses. Here's one, and here's all the chromatin. There, it's just in there. No particular organization. This is by chromatin, I mean chromosomes. Right? This is all your DNA. It's all just there. It's not organized. And then here, after the chromatin condensed, it's going to look like, I don't know, like DNA, like you think DNA looks like chromosomes look in the pictures. You get the idea. All right, so this is chromatin condensing. This is condensing, it's all coming together and getting like organized into like formal chromosomes. Because you can't split this apart. You can't split this in half. We don't know where everything is. Like it's not all equaled out. So anyway, the chromatin condensed, so it goes from like your nucleus looks like this before. At the end of interphase, it looks like this. By prophase, it's going to look like that. And in the lab, you'll see, like when you do the online lab, there's one lab that just shows you these different photos, so you're going to look at plenty of them. That's it. Those two things. The centrioles are going to start to move apart. The chromatin are going to condense. So I'll write my new, make my new nucleus here. I'll put like the whatever. There. All right, that's prophase. All right, so I'm going to erase this and I'm going to put metaphase now. But I don't want to write all of it on here because at the same time it's like a lot of words together. Let me erase this too. Can I? Oh, keep it up. All right. So let me move to metaphase. What happens in metaphase is that these centrioles have moved apart. They are like, they're on opposite ends. And these chromosomes are all lined up inside. So there's like an imaginary line that comes across, and they are all, and I can't draw these, but all the chromosomes are lined up. In fact, I drew them, whatever, there. So metaphase, what do I put? Chromosomes are lined up along the metaphase plate. So the chromosomes are all lined up across the metaphase plate. I want to redraw my chromosomes here. Because really they should be like that.
That's the metaphase plate.
Yeah, there's like an imaginary line that you would see coming down here. They're all, and so now they're kind of equal, like the chromosomes that are on the right side here are exactly the same genetically as the ones that are on the left side. So you can kind of see the line. You can imagine that line down the middle here where it's going to split. These centrosomes or centrioles, they're going to pull them apart. So they're actually, at the same time, they're putting out these rods, like these, or you can look at it as like a fishing line, like they're casting lines and they're attaching here and they're going to reel them back, right? So these are like the two fishermen and they're throwing out their fishing lines and grabbing on, and then they're going to reel it back and pull them back. So there's like these, we call these tubes. So I'm just going to draw them with black here. Some of them are attaching. I'm just going to draw it on this side, I guess. No, I'm going to draw it on both sides. There. So some of these, we call these micro tubes. Micro small, like micro tubes. Tubules. So we call these micro tubules. Some of them are attaching to the chromosomes, but some of them aren't. So you notice some of these are just missing altogether. So I got to throw in like this. I got to throw in this word that kind of sucks here. It's called kinetochore. And that's this, you see right here where the microtubules are touching the chromosomes? That region, kind of like, they look like X's, so like kind of where that X where it crosses, that's called the kinetochore region. So you have some that touch this part. And we call those kinetochore microtubules, sorry for the word. And then we have some that aren't, like over here. They're called non-kinetochore microtubules. So it just means some tubes are touching, some aren't. So chromosomes are lined up. We just leave it like that. The chromosomes are lined up along the metaphase plate. And I'll move to anaphase. Now I'm going to talk about these microtubules with anaphase. So in anaphase, you're going to have two things happen. One, see all these tubes, these microtubules that are attached to the DNA? The kinetochore microtubules, they're going to start to pull back now. They're going to shorten. So in anaphase, the kinetochore microtubules get shorter. Trust me, it's like worse. It could get worse. I'm just kind of giving you the minimum. So these are getting shorter. These continue to get longer. So let me draw my new chromosomes now, because since these are getting shorter, it's starting to look like they're getting pulled apart. So it looks like something like this. Now they're getting pulled back. So the kinetochore tubules are starting to retract, and that's pulling them back. So now they're separated. You have a nucleus, but inside the nucleus, all the chromosomes are kind of like on one side or the other side of the nucleus. And the non-kinetochore microtubules are going to, they continue to get longer, and they like push on each other, and they make the cell start to look like that, right? The cell's not round anymore, it's like oblong. You see like it's going to split right now. You see how it's going to split down there. So these non-kinetochore microtubules, they're pushing against each other and pushing the cell like that. So that's what happens in anaphase. Then finally you have telophase. And telophase, it looks something like this. In fact, I'm going to make a, I'm going to leave that. No, I'm going to erase the whole thing. Whatever. So there's telophase. Here's the genetic information, like the nuclei. The nuclei split. It's like you have two nuclei in one cell. So telophase. Nucleus splits. I'll put cleavage furrow here. Cleavage. Cleavage furrow is like, it's cleavage. It's like that. All right, so we call this a cleavage furrow. We can see now, like, this cell is gonna, it's gonna split. Take off mine. And there's a couple other things that happen during this, but this is essentially what we end up with. See these black, those black microtubules? Like, another term for that, I know I'm throwing like a lot of terms in here, but when you take like all the microtubules together, another thing we can call that is a spindle fiber. Spindle. That's just like another term. Like all these microtubules are called spindle fibers. Anyway, what are the things that I've been wanting you to know so far? Each time that I've talked about one of these, I've written one or two things. If you know that, you'll be okay. Those are the questions that I'll ask. So like what happens in each phase? It'll be something from what I've written here.
What's under nucleus splits? Cleavage furrow?
Cleavage furrow.
Furrow, okay.
F-U-R-R-O-W. And then some books, there's like a difference of opinion here. Some books consider telophase to be, so there's a third step, number 3. I have number 1, number 2. Number 3 is called cytokinesis. Cyto meaning cell, kinetic meaning movement. Cytokinesis is when one cell finally splits apart. Some people think that cytokinesis is part of telophase. Some people believe that cytokinesis is its own step. It really doesn't matter. They're both correct. I mean, because you have telophase, then right after you have cytokinesis. I count it as its own step. Because sometimes you can have telophase finished, and you have one nucleus with two, you have one cell. So sometimes if you look under a microscope, you can see one cell and there's two nuclei in it, and it didn't split. For some reason, everything stopped right at that point. So we didn't finish. We didn't end up with two cells. But I mean, who cares, right? It's not going to be on the test. Cytokinesis is number 3. So interphase, mitosis, cytokinesis is the cell splitting, the cell itself breaking in half. It's really an easier process that I have made very difficult by breaking it down and talking about each part. If you just look up some videos and you slow it down and watch the cell break in half, then you're going to be like, oh yeah, I totally get it. That's so easy. And then you'd be like, why did he have to talk about each part? Well, that's the question. Why then? Why do we have to make a whole chapter on this, Rod? Like, why do we have to take a nucleus breaking in half and make it so long? Right? So I'm going to erase all this now. Because when you study cancer, cancer is the cell cycle without any controls. I don't know. Like our cells divide, right? And we know they divide, but they don't divide forever, right? We think that there's like built into our cells are like time limits, right? Like these cells don't divide for 200 years, right? We die. And so we think that we call it apoptosis, that there's this fixed time built into our cells, and when it reaches that, they're gonna die. But also, you can't have every cell divide all the time. You gotta have like some kind of stop, right? And my hair is going to stop going through cell cycle eventually, right? All your cells, so when do you do it and when do you not do it? I mean, we don't really know. We have had a couple ideas about what stops it. And so one idea that, like, why do cells stop dividing at a certain point? It's this idea of density dependency. I got about 5 more minutes left, just so you guys know. We'll be out of here. Density dependency governs the growth in our body. It also governs everything. It governs nature. You don't have, if you have too many coyotes, then you don't have enough rabbits because all the coyotes are eating the rabbits, and then the rabbit populations plummet, right? And then there's no food for the coyotes, so the coyotes start to die off. And when the coyotes die off, the rabbits start to go up. And when the rabbits go up, now there's more food for the coyotes, so they're kept in a balance. Density dependency. You can't have too much. Humans are the only ones that can screw this up. We actually, I guess we're kind of like a cancer in that way. We ignore rules of density dependency. But nature works with this density dependency. But humans do too. If you get too many humans together and you're eating bats, maybe you start getting diseases and dying off, right? Too many people. But anyway, so our cells work on this idea, right? And then there's also other chemicals. There's chemicals, and then there's like checkpoints. For example, if you remember, like if you look at the notes on interphase, I put G1, S, G2. There's checkpoints in there that your body will follow. Like if things aren't right in the cell, your body will not go from like G1 to G2. Or your body won't go from S phase, from like anaphase to telophase. Whatever the phases are, we have like checkpoints that our body will check. All right, does everything look good? Do we really need this cell to divide? Okay, then keep going. But cancer ignores all that, and it just keeps going. It's the cell cycle, just division, 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 and if you get too many cells at a point, then you end up with a tumor. And so I guess there's two states of cancer that you can be in with cancer. That one is like benign. That even is how you spell it. The other one is called metastasis. That's kind of like the important word. And metastasis is when this tumor, if some of the cells in this tumor break off and get into your blood or your lymph. We call these neoplastic. Cancer is like a neoplasm. Just another word. Enter blood or lymph. Lymph is another, it's another way. If blood is like the freeway system, lymph would be like the service roads, right? It's another way to move around, stuff around the body, right? So you have like blood vessels coming around your shoulder. You also have lymphatic vessels coming around your shoulder. We just don't talk about it. So benign, let's say you have like, breast cancer, and then there's a tumor that forms in your breast, but the tumor didn't grow enough until it got to a blood vessel. So it didn't get into your blood. That would be like the better choice, because you can cut that out and then just watch the person and hope that it didn't like spread anywhere. But if that tumor grows and it gets to a lymphatic vessel or blood vessel, then those cells, once they enter the blood, they can end up anywhere in your body. So now you have to take a different approach to this. You have to now try to like nuke the body and kill off the cancer cells. And so the idea behind radiation or chemotherapy, especially chemotherapy, a lot of chemotherapy agents, they stop mitosis. They arrest different stages of mitosis. So chemotherapy, we call chemotherapy cytotoxic. Cyto means cell, right? So chemotherapy agents are cytotoxic. You're killing all your cells. And you're like, well, even good cells? Yeah. Good cells, bad cells, you're just attacking the body and you're killing everything off, and you're hoping to kill off the cancer, and then you can pull back the chemotherapy, and hopefully all the good cells will start back up again. And that's like a way to do it. So how do we kill all of our cells? How do we stop them from dividing? So you stop the spindle fibers, for example. One type of chemotherapy will stop those microtubules, right? So if you can stop those tubes, then they don't grab onto the chromosomes, and they don't pull them apart. You don't have anaphase, you don't have telophase, right? So they all target. So when you learn about chemotherapy agents, you're going to study mitosis. And so some of them attack that, some of them stop the metaphase plate, so all the chromosomes can't line up, they're just all disorganized, and because of that, you can't break the cell in half. So when you learn about chemotherapy, it's all about like mitosis, or if you could stop a cleavage furrow. So anyway, metastasis is when it spreads. All right, so any questions on the cell cycle? Definitely know what metastasis is. Have an idea what cancer is, of course. Interphase, I don't care about G1 and G2. They're the same thing, actually. It's just called gap 1 and gap 2. But you should know that during an interphase, the S phase of interphase, that's where your DNA is made. So I sometimes will ask a question about that. In mitosis, you've got the 4 stages. Your book might say pro-metaphase. That's a 5th stage. You could ignore that. I might ask a question about, like you should know what happens in prophase, metaphase, anaphase, telophase. You should know what cytokinesis, what does that mean? The cell is splitting itself. Here's the very last thing that I want you to know for the test. All of this that we were talking about were eukaryotes. There are 2 types of cells, eukaryotes, prokaryotes. We will not be talking about prokaryotes because they're just bacteria, pretty much. They're a different type of cell. They don't have a nucleus. I mean, this you should have already studied. Prokaryotes don't have a nucleus. They have what's called a nucleoid. And so, since they don't have a nucleus, we don't have mitosis. We don't have the same, it works on a different system. And so that system is called binary fission. I don't even know how to spell it. Hey Siri. Binary fission. See, Siri can't answer all your problems. Whatever, I think that's it. So binary fission, that's how bacteria divide. They use a different system, and it's really simple. I didn't even bother teaching it to you because it's really easy. And that's what bacteria do. They divide, that's it. That's their reason for being. So prokaryotes, which are bacteria, use binary fission. That's it, we're done for that. Eukaryotes are every other type of cell. Animals, plants, fungus, fish, whatever. They're all eukaryotes. The only things that are prokaryotes are bacteria, and then there's these things called archaea. If you have to take microbiology, then you're going to spend all your time on that. So I might ask about that, like what do prokaryotes or bacteria, how do they divide? They use binary fission. And all they do is they make a bunch more DNA and they split in half. There's no organized process for that. All right. Any questions? So what we're missing is respiration, cellular respiration. So let me stop this lecture. Let me stop recording.